Okay, so we're going to start today with just one section of the textbook, but we're going to go into a little bit more detail than what the textbook actually has. So when you do go to study this chapter for the exam, just make sure that you actually study the PowerPoint material. Obviously, not the exam today, but the exam 2 weeks from now for this chapter 7.3, section 7.3. You'll just want to make sure you review the PowerPoint because the book is really vague and there's just a lot of interesting details that we'll kind of go over today. Nothing too complex, just some disorders that the book doesn't really talk about, but that are kind of good for you to be aware of that they exist. So if we have chromosomal aberrations, we talked in chapter 7, in 7.1 and 7.2 about cell division and proper cell division and what that looks like. We talked about mitosis, which is what all of our body cells and our skins and our organs, our hair, your mouth, everything in your body, but your sex cells is going to be, 2 of each chromosome normally. And so we talked about mitosis and how we have to have replication of the DNA and then just one division process where we have 2 new cells, each of them with 46 chromosomes at the end of that process. And then we have meiosis, where the same thing happens, but it happens twice. So you get sperm and egg cells that are haploid. They have 23 chromosomes in each of them. So we take our one copy of DNA, we replicate it, but then we end up with only haploid cells at the end of the day, so that when they go through fertilization to produce a new individual, that individual still only has 46 chromosomes. There can be issues in meiosis, especially. Think about it. You have 2 processes of division. So you're going through prophase, metaphase, anaphase, and telophase 2 times. Not really weird, when you replicate your DNA and then both of the chromosome 17s line up on the metaphase plate. And so we have what we call tetrads. And then we have some crossover events where literally some chunks of the chromosomes are being swapped with the other chromosomes next to them. It is somewhat dangerous because you can have those swapped segments ending up in the wrong places. You can have tetrads that instead of separating and going into 2 new cells, they don't separate properly and they just go into 1 cell. So we're going to look at the consequences of things like that happening today. And all of those are chromosomal aberrations, basically mistakes in the chromosomes. So aneuploidy, diploidy would just be like us, where we have the proper number of chromosomes. Aneuploidy is where you have an alteration. So monosomy is when you only have 1 of a chromosome, and trisomy is when you have 3 of one of the chromosomes. And we'll talk about some diseases or some conditions, I suppose would be the best word, where you can actually have 3 of one chromosome, but actually survive and be a whole human being, but obviously have some potential issues because of it. If you are polyploidy, this is really bizarre. This would be not having 23 or 46 chromosomes, but whatever 46 plus 23 is, so 69 chromosomes total in all of your cells. And humans cannot survive with a whole extra set of chromosomes. We have some organisms on the planet that are a lot simpler, I guess you could say, in terms of their DNA. They don't have 23 chromosomes like us, so they don't have as much genetic information. So there are a few organisms out there that can survive as a polyploidy individual, but in humans, it can't happen. We can have a couple instances of monosomy or trisomy and survive, but you'll have some serious issues with that individual in normal development won't really occur. So non-disjunction in meiosis is how these things happen, where you have one of those tetrads that is not separating properly. And so you have some of the eggs or sperm ending up with too many chromosomes and some of them ending up with not enough. And both of those cases would cause problems. So 20 to 50% of spontaneous abortions. Obviously, this is really hard to know exactly how much of this causes spontaneous abortions, because a lot of times mothers don't even know they're pregnant when they have an early miscarriage. And so spontaneous abortion just means that your mom's body is doing it herself, or the embryo is basically causing abortion itself. And so that can happen in 2 ways. If mom if her body's not developing properly, she doesn't have enough hormones, or her uterus is not developing as it should, that can cause a miscarriage to occur. But it can also occur in the embryo itself. If that embryo had non-disjunction events happen in meiosis, in either the egg or the sperm before fertilization occurred, and things didn't work properly to create a new individual, then a lot of times that embryo will kind of abort itself, essentially. So what does this look like? Let me just remind you, since it's been a while now since we learned about meiosis and mitosis, the regular cell division stuff. So meiosis 1, if you have regular events occurring and we have no issues, what we would start with is, maybe this is chromosome 16 and 17. So we would have 2 copies of those in meiosis 1, because that had been replicated, and you want to have 4 daughter cells at the end of it. So in meiosis 1, they're going to line up at the metaphase plate, but those tetrads are going to split apart into 2 new cells. The tetrads will then, after the tetrads have split up, the chromosomes will split into their sister chromatids. And what you should get at the end of it is 2 cells that look like this, or 4 cells that look like this. But they're showing you all of the non-disjunction events that could happen. So you can have non-disjunction where that tetrad is not breaking up in meiosis 1. And so you have 2 of the daughter cells that have 2 copies of chromosome 17, but then you have 2 daughter cells that don't have any copies of chromosome 17. So that is a pretty major non-disjunction event occurring that is basically creating a trisomy individual and an individual that's not even monosomy. They don't even have 1 copy of that gene. So obviously that's going to create some issues. If this egg was fertilized by a sperm, then the zygote would have 1 copy of chromosome 17, we presume, from the sperm, but it could still create some problems. So the other way that non-disjunction can happen is in meiosis 2, where you would have one of those chromosomes, again, the sister chromatids not separating. So here's what it looks like when it happens correctly. But if the sister chromatids don't separate, you get another scenario where you have 2 chromosome 17s in one egg, 0 chromosome 17 in the other egg. Once these eggs are fertilized, this egg would have 3 chromosome 17, this egg would only have 1. So you would have a monosomy or a trisomy individual, possibly from that one non-disjunction event. So non-disjunction, I mean, usually when it occurs in like chromosomes, like our normal body chromosomes, we do have sex chromosomes like an X and a Y, which you've probably heard of. And females have 2 copies of that X chromosome. Males have an X and a Y chromosome. Most of the conditions where we have non-disjunction events happening, the individuals that survive, it probably occurs within their sex chromosomes. And that's just because there is not as much genetic information on those chromosomes. They're not as big, so there's not as many genes in them. If this happens on like chromosome 1, 2, or 3, which are our biggest chromosomes, the individual is likely to not survive because there's too much genetic information, too many genes being messed up in that scenario. Okay, so this little diagram, I think, can be a little bit confusing at first, but female non-disjunction. So if we have an egg where meiosis does not happen properly versus a sperm where it doesn't happen properly, we can get different results because females have 2 X's and males have 2 Y's in their body. So if we have an egg that didn't go through disjunction properly, and so we had one of the daughter cells had 2 X chromosomes, where they each should only have 1, and the other daughter cell would have 0 X chromosomes because they both went to this cell, we can have 4 potential outcomes of this. If you don't have any X chromosomes, you cannot survive. So that embryo would not survive. It would go through spontaneous abortion pretty early in the pregnancy, most likely. Even after being fertilized, gets a Y chromosome from dad, but there is no genetic information needed from the X chromosome because we don't have an X chromosome. And there are some traits that you need in order for development to progress. Turner syndrome, Klinefelter syndrome, and trisomy X, all of these have names because individuals with these conditions can survive. And so we'll see what these look like. But if you had this egg that did not go through disjunction properly, so non-disjunction, both X chromosomes were passed into this egg over here, fertilized by a sperm with an X chromosome, because dads can donate either an X or a Y on their sperm because they have both X and Y chromosomes in their bodies. Trisomy X is when that offspring has 3 X chromosomes, 2 came from mom, 1 from dad. XXY is if they got 1 Y from dad and 2 X's from mom. Turner syndrome, last one for the female side of things, if the egg does not disjunct properly, would just be 1 X chromosome, no Y chromosome, which you would think maybe would be okay, but you really do need 2 copies of every chromosome to have proper development happening. And you can kind of think of it as like the X chromosome is responsible for like female hormones, and you need enough female hormones in your body as you develop to have proper development occurring in the growing baby and even when they become an adult. And so if you don't have enough of something, it can still lead to problems. And I think that I fixed this. I think I had it wrong on a PowerPoint on Canvas. This should have said sperm up here. So if you want to change that really quick, you can do that because we're looking at male non-disjunction. So if disjunction does not occur in the original parent sperm cell, we want to know what the issues could be there. And you should see that 3 of these syndromes down here are exactly the same as they were on the female end. But no matter what, in all of these cases, if you have non-disjunction happening in a sperm cell, you will get a viable offspring. They are less likely to survive, but all of them potentially can survive because they all will be getting an X chromosome from mom. The X chromosome is a lot bigger than the Y chromosome, so it's not as big of an issue if you don't get that Y chromosome from dad for some reason. It is still a big issue, but you can survive potentially as an individual with only 1 X and no Y. So we're just going to go over a couple of these syndromes, and I'm not going to spend a lot of time on these. When you study for the exam, it'll just be, some pretty basic memorization of kind of what these are, and there won't be too many details on it. Turner syndrome is the one where we have 1 X from mom or from dad, but then we don't get a Y chromosome or a second X chromosome. So just X0 is what they call that, or XO. So you only have 1 sex chromosome. They can survive, but a lot of times they do die before birth. It just kind of depends on how the development process goes in the uterus. But hormone deficiencies, yes, always, because you don't have enough hormone production, because you don't have enough of that female hormone being produced, or you don't have any male hormone being produced if you were an XY individual. So sterility in a lot of these conditions is very common. Secondary sex characteristics, so you can think of that as just being a result of hormone deficiencies. If you don't have enough estrogen and progesterone in your body and you're a developing female, you won't have breast development, and you probably won't have, pubic hair and any of those secondary sex characteristics. Menstruation is just kind of part of that. Usually short in stature as well. A lot of our sex hormones, especially testosterone in males, both male and female people produce testosterone. Males obviously just produce a lot more of it. But if you're lacking sex hormones, you can have shorter stature because of that. The extra skin on the neck, these are just kind of common physical characteristics that would help you determine what condition this person has. And then, a variety of other health problems. So this one, normal intelligence, the difficulty with spatial and math skills, which I think kind of plays to the stereotype that men should be better at math. But it is a pretty common characteristic of someone with Turner syndrome. Fairly normal intelligence, but just issue in those couple of areas. Trisomy X. So anytime you see trisomy, it means 3 of a chromosome, and the X is just which chromosome it's referring to. So trisomy on the X chromosome means you have 3 X chromosomes. You can be infertile. This not necessarily, because if you have enough of the female hormone being produced, you would think that you could go through puberty and all of that stuff. But having multiple copies of a chromosome can still kind of cause problems with normal development. If you think about cells going through cell division, there could be mishaps there during development as well if you have an extra chromosome that you're trying to deal with. And these individuals would usually be taller. So anytime you have extra chromosomes, you can kind of think of it as extra of everything. Klinefelter syndrome. This one is a little bit more common than the other, so 1 in 1,000 births, and this would be an XXY chromosome. So a little bit of like a combination of male and female characteristics in the physique of these individuals. It is strange that they're taller than average, I think, but if you're trisomy X, you're also taller than average, so it kind of makes sense. But the major things in a Klinefelter individual is that they'll be sterile, and then they'll have some breast development usually. Females usually have some fatty deposits in the abdomen and on top of the pubic bone. So you'll see some fatty developments there where females would normally have it. Usually the testes are smaller as well. And this is mostly because you have more female hormones being produced. And so those are disrupting the pathways of normal male development. So a lot of this would be causing issues during embryonic development, creating those pathways, the wiring in the brain needed for male effects later, but also during puberty, this could cause issues as well. So reduced body hair, that's because you have fewer of the male characteristics, then you're also having some of the female characteristics develop as well, because it's kind of a combination of male and female chromosomes. XXXYY, also a little bit more common, so just as common as Klinefelter syndrome. Undetected because the Y chromosome is so small that I just don't think that it really disrupts that much. So it says 1 in 1,000 people. It's possible that it's more common than that just because it's not tested that often, because these individuals wouldn't really have any issues normally in their appearance or in mental capabilities. If we have autosomal non-disjunction, so this is a more serious case, and so there's not as many chromosomes that are going to go through non-disjunction and produce a viable embryo. Like the chromosomes 1, 2, 3, and 4, like the larger ones, if you have too many or not enough of those chromosomes, that individual probably won't make it past, you know, the early stages of embryonic development. But we have a few cases where they can. So Patau syndrome, which is trisomy 13, which means you have 3 copies of chromosome 13. And a lot of these conditions, they will die as an infant just because that really creates some problems when you're replicating these larger chromosomes. Edwards syndrome, which is trisomy 18, a little bit more common, but again, some pretty major issues a lot of times. Extreme muscle tone. So trisomy 18 is the chromosome that I believe codes for your proteins in your muscles. So every single gene codes for a protein, because that's what, you know, transcription and translation, when we went through all of that, that's what they do. But if we have more actin and myosin, the proteins that make up our skeletal muscle, obviously that will lead to more muscle tone because you're just producing more of those proteins. But so many of these chromosomes just have genes that code for things, for proteins that work all over your body. So usually the symptoms will be very, very widespread, and usually some mental issues as well, because they're going to, you know, mess with developmental processes in the brain. Down syndrome is probably the most common, and I think most likely to live longer and have, you know, the most normal life of any of these non-disjunction conditions for the autosomal non-disjunction ones. So the issue, and, you know, why we talk about females, you know, why it's risky to wait to have kids until you're older, and why some females opt to freeze their eggs. I mean, I know a lot of like celebrities do that, a lot of female celebrities, because they want to pursue a career first, but they still want to have kids later. One of the reasons why it's good to have kids when you're a little younger, or, you know, have your first kid at least when you're a little younger, is because some of these conditions are more likely with an increasing age of the mom. I know somebody with a condition, basically dwarfism is the layman's term for it. That condition is more likely to occur when the father is older. So it's not always an issue of increased age of the mom, but that is oftentimes associated with Down syndrome cases. So the lifespan is definitely going to be limited, but they can live a pretty normal life. You probably already know somebody or have met somebody with Down syndrome, so you know a lot of these common physical characteristics, so I won't really go through them here. Health problems, mental issues are very common, developmental problems that just kind of result in limited mental capabilities, I suppose. Any of these things are a result of big issues on chromosomes, where you have a whole chromosome not splitting properly in the tetrad state, or the sister chromosome splitting in meiosis. But we can have small scale issues as well that would probably lead to a viable individual, but it could lead to you having a certain disease or a certain digestive problem or a certain problem with one of the organs in your body if the gene that codes for a bunch of the proteins that work in that organ, or if you are missing the gene that codes for an enzyme that digests a certain food group, obviously that could still lead to problems. So these smaller scale alterations can still lead to major issues. It kind of just depends on where they are on the chromosome and how important that gene is. So a deletion is just when you delete a tiny portion that doesn't get sent to the to the daughter cells at all. Duplication is when you would end up with extra, and can cause issues, but it would probably be less likely to cause a problem if you have extra of a certain chromosome. The order of genes are important because DNA synthase and all those proteins that are involved in replicating DNA, they have to have their start codons and stop codons. They need to know the exact order that all the genes are going to be in. So if you have an inversion occur, it's possible that those genes won't ever be transcribed because those proteins and enzymes don't know where to find them. And then translocation, these are probably most common, definitely common in the tetrad position. So the tetrads is when you have like chromosome 17 from mom and dad that have been replicated. They both line up in meiosis 1 on the metaphase plate, and then they exchange some of their bits of chromosomes. But if you have exchange of the chromosomes happening and it doesn't happen correctly, translocation or an inversion or any of these things can happen as a result of that. Cri du chat or cri du chat, it's actually a French term, so cri du chat I think is the proper way to say it. It's also called cat cry just because of the noise these individuals make. It kind of sounds like a cat crying. So cri du chat is just the French term for cat cry. So deletion of just part of chromosome 5, and most of them don't even make it past childhood. So if you delete a part of one of your major chromosomes, you can think of a deletion as being a pretty minor issue. You're just taking out one chunk of that chromosome. But if that chunk codes for something that helps your neurons develop or your heart develop properly, that can obviously cause major issues. And all of these things, like I mentioned at the beginning, I just put a slide in here at the end to remind you, these are definitely a major cause of spontaneous abortions. So if you know somebody who had a miscarriage really early on, maybe it's not because of genetic mutations, but it's a really a safeguard to prevent individuals who wouldn't be able to survive. Even though we do have a lot of these conditions occur, most of them would probably go through spontaneous abortion early on in development because you just wouldn't be able to survive with those conditions. Alrighty, we'll go ahead and move on. So you probably, it maybe has been a while, but you've probably covered genetics in some form. I think I did this in middle school, some Punnett squares and stuff. So a lot of this unit will be more comfortable for you than last week. And so the exam you're taking today, in my opinion, is probably the toughest exam in terms of content for the course. I think that we'll start to get to stuff that you're more familiar with and that is more easier to conceptualize because you can imagine these things occurring in real life, whereas you can't really visualize the chemistry stuff and the bonds and things like that because you can't really see them with the naked eye. So hopefully this feels like a little bit of a review for you today. But I will go through the PowerPoint now, and then we will do a few review questions. So if you want the full 10 points today, hang around at the end. We'll probably spend like 15 to 20 minutes doing a few review questions, just because you can't really gain much from me talking to you about Punnett squares. You just have to kind of do them yourself to see how they work. It's just one of those things that's easier to learn when you're doing it. I'm going to put up this slide and just stay here quiet for a couple minutes and give you some time to fill this out. So the red things, I just thought you'd be less likely to know, which is why I'm having you fill them in on your own. So I'll give you a minute. So all these terms maybe sound familiar, maybe you don't exactly remember what they mean. I'm not going to go through them all on this slide, but we'll talk about them all throughout this PowerPoint, but you can always just come back to this slide if you forget what any of these things are. I think the most confusing one for any student that I have in any class where we learn about this type of stuff is like the locus and the allele thing. So you have genes that code for proteins. An allele is a gene, but it's just a different version potentially of a gene. So an allele is a gene, but you have, multiple alleles that can code for one gene. And so the easiest thing to think about when you're trying to remember what an allele is that if you have flowers in a certain species of flower that can be purple or white, one allele would code for purple and one allele would code for a white flower. So both of them are genes, they're just different versions of those. And I hope that term makes a little bit more sense at the end of today. And the locus is just where on the chromosome that allele is. So if we have homologous chromosomes, and you can imagine these being within the cell, as we go through the replication process, it's important that these are able to line up perfectly with each other. Part of that is because when you go through meiosis, you want to make sure that you're exchanging the proper bits with your neighbor chromosome. And that's important because the genes that code for certain proteins will always be on the same spot on those strands of DNA, right? And so when we have chromosomes, you might have a certain gene that codes for purple flowers or white flowers. You'll have another trait, another gene, another portion of that chromosome that can code for the color, for the shape of the flower, so on and so on. So we'll look at how this all works in Mendel's studies, which is what, usually you probably went over Mendelian genetics in middle school and high school as well. And so this should start to sound kind of familiar. So locus is just where it is. An allele is just a different version. So these two colors down here are showing you that there are two separate alleles that code for that same gene. One comes from mom, one comes from dad, but you can have both of them. You can have two separate alleles in your body at any given time because you do have two copies of each chromosome. So Gregor Mendel, and just, there are some really important scientists in history. And so I know it sounds silly to try to memorize names for exams. This is really the only one that we'll have for this exam is Gregor Mendel. But he's really what we call the father of genetics. Like he is the one that defined basically all of these things. And he didn't even know like what chromosomes were, which is kind of amazing that he was able to kind of figure out all of this stuff without even being able to see these things in the cells because he just didn't have that equipment yet. So the reason that he wasn't taken very seriously is because we couldn't really see how this stuff actually worked in the cells themselves. And so like several scientists throughout history, he probably didn't even really get much praise for his work until after he died, which is kind of sad. But all of this stuff that he did for genetics, creating these, terms that we know today, he did with pea plants. And so we'll use those as examples in this class because it's really, really simple to use them because they have such simple traits where they're either purple or white and there's really no in between. So what he did in each of these steps is that he crossed breeding lines that have the same phenotype, Phenotype is just what the characteristic looks like. So you have genes that code for hair color. Your phenotype is whatever hair color that you have. And hair color is obviously a weird one because it can change over your lifetime, or it can be in between your mom and dad, but different phenotypes for the same trait is what he was focusing on in all of his studies. So he took pea plants, and pea plants always have flowers because they're an angiosperm, and angiosperms always have flowers, but pea plants are pretty simplistic in a lot of ways. So he looked at a lot of traits: flower color, the shape of the pea, the height of the plant. And in each case, they're really just one or the other, like they're purple or white. They are smooth or wrinkled peas. They are tall or short. And so it's a very simplistic species to use for this type of stuff, which I think he went through and tested other species, and he found that this was kind of the best one to work with. And so he basically controlled which peas bred with which peas. And so he very much controlled all of the reproductive events that were happening in all of these cases. So a little bit of new terminology maybe. So the F1 and F2 generations just means first offspring and second offspring. So if we look at this example where Mendel crossed purple and white flowers, we have a couple of key things that we need to kind of identify on this little diagram over on the side. So he crossed purple and white plants, and that was his parent generation. He then observed that all of the F1 generation were just purple. He did this several times, he did this like thousands of plants in the end. Every time he did this cross up here, we'll talk about what this is, he got all purple offspring. He then cross-fertilized or self-fertilized this F1 generation. So it's important that we kind of recognize who's the F1 plants. Self-fertilized the F1 population, and he got white offspring from those all purple F1 generation, which tells us that somewhere in this purple plant, they still have the trait for the white flower. It's just being covered up. So if you remember the terms homozygous and heterozygous and indominant and recessive, that's where these things come into play. And so this parent generation that he crossed, this purple plant right here, had two alleles. Both of them were purple. This white plant had two alleles. Both of them were for a white flower. And so we call both of those parents homozygous because they had the same allele for each of those genes on their chromosomes. The F1 population was cool because it's going to be a heterozygote, which means it had a purple allele and a white allele on each of those two chromosomes that code with the genes that code for flower color. So if you cross heterozygotes in this F1 generation, every single time you do a cross like that, you will get 75% of one of the dominant trait and 25% of the recessive trait. And so we'll kind of look at this in a lot of different traits that Mendel tested this on. So the seed color, yellow versus green, shape, the pea pod and the pea pod color. All of these tests that he did ended up with almost being exactly 75% of the dominant trait and 25% of the recessive trait in the F2 generation. So obviously it's not perfectly a 3-to-1 generation because not all of the seeds are going to germinate, but if you kind of average these out, they would all be approximately 3-to-1. So he got the same exact ratio. And he didn't even know kind of why this was happening. He was really the first person to actually do this type of test. But what this kind of gave us was a few different laws. And so the first one is the law of dominance, which if we look at the purple and the white flowers, if you end up getting purple flowers only in the F1 generation, that means that your purple color flower is going to be dominant over the white. It doesn't mean that the white allele completely disappears in the F1 generation. It's just being covered up. And so blood type's weird, and I'm O. But what that means for me is that I have two recessive alleles for type O blood type. And if Michelle is AB, that means that she has an A allele on one of her chromosomes and a B allele on the other one. So not all traits, just like blood type, not all traits are going to be this simple dominant and recessive combination, but that's really what we're going to focus on in here. So in blood type, A and B are co-dominant, so you can express A and be an AB blood type. You can't be purple and white for these flowers. You can't be somewhere in between. That's possible for a lot of different traits, but everything that we're going to be looking at for the Mendelian genetics is just simple dominance, where one color is dominant over the other. So it's all of this F1 generation has both alleles, but the white allele is just being totally covered up by the purple one. In other words, if the purple one's there, it's the only thing that's going to be expressed as the phenotype. So the important things to note is that if the parent only makes one type of gamete, like they only have one possibility coming from them, they are homozygous. So this purple flower up here in the parent generation, big P, they're homozygous dominant. The other parent is also homozygous, but they're homozygous recessive. They have little p, both coding for the white flower type. Everyone in the F generation, because they got one allele from the purple plant and one allele from the white plant, every single individual in this F1 generation is going to be big P, little p, which makes them a heterozygote. Heterozygous just means you have a combination of dominant and recessive alleles on your chromosomes. The F2 generation will be kind of a mixed bag of homozygous and heterozygous. There'll also be a mixed bag of colors, the phenotypes coming from that. But you'll have more purple ones because anytime you have that dominant allele, the flower is going to be purple. There's only one scenario where you can have a white flower, and it's if you have two of the little p allele. So if we have a simple dominance scenario, which is what we'll always talk about for today, where you just have two alleles coding for a trait like flower color, and you have just a dominant or a recessive trait, you'll always end up with this ratio of genotypes and phenotypes, where you'll have one homozygous dominant, two heterozygotes, and one homozygous recessive, and that will result in 3 purple flowers and 1 white flower. So the genotype is just what you get as the result of your Punnett squares. The phenotype is what those things actually look like in real life. And we can see, kind of break that down a little bit more, so you can kind of go back to this slide and remember, what does homozygous mean? What does heterozygote mean? And then what does that mean for their genotypes and phenotypes? So the phenotype is just what that looks like in real life. So your phenotype for hair color would just be what your hair looks like. The genotype is what genes or what alleles do you have that create that hair color. Okay, a test cross is what Mendel did in all of these things that we've already looked at so far. A test cross always starts with homozygous parents. If we want to be able to go through this, parent F1, F2 generation and get that kind of perfect 3-to-1 ratio at the end. So we can also use a test cross, though, to determine the genotype of an unknown parent. If we have a parent who is purple, but we don't know if they are big P or big P, little p, we can go through a test cross them with a homozygous recessive individual. And just by looking at their offspring, we can figure out, was that parent big P, or were they big P, little p? So if they're big P, you're going to get all purple flowers in the F1 generation, their offspring as a result of that cross. If this parent was big P, little p, and you cross them with a little p white flower, half of the offspring will be purple and half of them will be white. And I know this might be a bit confusing at the moment, or you remember it, but you don't remember how to visualize this stuff. We'll go through some Punnett squares at the end, so don't be too worried about this at the moment. But this would be crossing a dominant phenotype with a recessive phenotype. And the important thing there is the recessive phenotype, because that allows you to discover the genotype of the other unknown parent. And that's just because you know that the white flower has to be little p, little p. There's no other way that you can get a white flower. Okay, the law of independent assortments. We have quite a few laws in this chapter. And remember that laws are just something that we've tested so many times and gotten the same thing again and again and again in a bunch of scientific studies. We've gotten the same thing so many times that we're pretty positive that it's actually correct, even though they're still kind of hypotheses. We're so sure that they're correct that we start calling them laws. So the next law is independent assortment, which means that different traits can be inherited separately of one another. So just because your mom plant had yellow and smooth peas does not mean that the yellow and smooth have to stay together in all of the offspring. So if mom was yellow and smooth, dad was green and wrinkled, you can have a combination of mom's traits and dad's traits in the offspring. They don't have to be inherited together. So you won't have offspring that are yellow and smooth or wrinkled and green. You can have yellow and wrinkled. You could have green and smooth. So that's what this Punnett square, big Punnett square down here is showing you. If those traits had to stay together, you would get, 3 individuals that looked like mom and 1 that looked like dad as a result. Basically like the simple dominance with the purple and white flowers that we just went through. There are some exceptions to this. If the traits are really, really close to each other, like if both of these traits were on chromosome 7, I don't know how many chromosomes plants actually have, but if they were both on chromosome 7 and they were right next to each other on that chromosome, it's possible that they're actually linked, which means they need to stay next to each other all the time for that chromosome to function properly. So when you go over, you have crossover events happening in that tetrad state, those would always be exchanged together. So there are some traits that would be inherited together, but for the most part, all of the traits that Mendel looked at can be inherited independently because they're on different spots of oftentimes different chromosomes. Okay, I'm just going to show you this slide, and I think the next thing is practice problems. So I'm just leaving this slide here for you kind of as a reference for you to come back to, because next, I think on Sunday of next week, you'll have a dihybrid cross assignment due, and it'll look a little bit more complex. And so it's really just helpful to have an example of how to set up these Punnett squares. So a dihybrid cross is just when you're testing 2 traits. And so you'll have a 4x4 Punnett square like this one.